Welcome back to another video with Miss Z. Let's start with a quick question. Have you ever wondered how your body knows how to make insulin after you eat? Or how do your muscles build back stronger after an exercise? Well, today, we're going to talk about a tiny but mighty structure inside your cells that makes all of that possible. The ribosome. By the end of this lesson, you'll be able to define what ribosomes are and explain the differences between free and bound types, describe how ribosomes help turn genetic information into proteins through translation, identify ribosomes in prokaryotic and eukaryotic cell diagrams, and finally, analyze what could happen if ribosomes stop working properly. So what exactly are ribosomes? Ribosomes are tiny cellular machines that build proteins. That's why we call them the protein factories in the cell. They read instructions from your DNA and build proteins your body needs to survive. Now, let's look at these two images. One showing free ribosome and the other showing bound ones. What do you notice right away? What do you see that is different between these two ribosomes? Exactly! One of them is floating and the other is attached. Free ribosomes float freely and make proteins for use inside the cell. However, bound ribosomes are stuck to the rough ER and make proteins for export or for cell membranes. Now that we know what ribosomes are, let's see them in action through translation, the process of making proteins. Just to be clear, the process called translation is not like translating English to Filipino or Spanish back to English, but converting mRNA into proteins. And these has steps. Number 1. Quick question. Who remembers where mRNA comes from? Bingo! mRNA is the messenger. It carries instructions from DNA out to the ribosome. Moving forward, step 2. Each tRNA brings something special to the ribosome. What do you think that is? Correct, as you see from the slide before, amino acids. Each tRNA has an anticodon that matches the mRNA code, bringing the correct amino acid for the growing protein. Lastly, step 3. What do you think happens when more and more amino acids join together? What did you say? A protein? You're on fire! They first form a polypeptide chain. The ribosome links amino acids together using peptide bonds and boom, a protein is born. Let's put your observation skills to the test, shall we? Can you spot the ribosome in each diagram? Point to it or describe what they are. In the cytoplasm on the rough ER in the eukaryote. Nice job. So even though prokaryotes don't have organelles like ER, they still have ribosomes in the cytoplasm. Ribosomes are universal. Why, you might ask? Because every living thing needs to make proteins. How about we think critically for a moment? What might happen if the ribosomes in your body stopped working or made mistakes? That is indeed correct. Without working ribosomes, the cell can't make the proteins it needs. this can lead to diseases. Basically, no ribosomes means no proteins means no life. For example, a rare disorder called Diamond-Blackfan anemia is caused by ribosomal defects. Also, did you know some antibiotics kill bacteria by blocking their ribosomes? Now that's a good fun fact. Let's have a quick recap, shall we? So these are the key points that we tackled earlier. Number 1, ribosomes are essential protein factories. Next, free ribosomes make proteins for use inside the cell. However, bound ribosomes make proteins for export or membranes. Next, protein syntheses involve mRNA, tRNA, and ribosomes working all together. And lastly, if ribosomes fail, there's serious problems. Now, I'd love to hear from you. What is one new thing you learned today about ribosomes or protein synthesis? Feel free to say it out loud right now or type it in the comment section if you're online. Great work today! Let's move on to a short quiz or reflection activity to see what we've learned. We'll be presenting a QR code here for a Kahoot, which is shown right over here. Quiz time!